everyone, and welcome back to Voices of Impact, a lovely project intended to bring people closer to the research that is being going here, mainly here in Europe. And not only that, but how our research improves and impacts on everyone's life. So my name is Mauricio, and as always, I have the honor to be the host of this beautiful video series. Today, we have with us our fifth guest, Ornela Urzì. So Ornella got both her undergrad and master's degree in medical biotechnologies and molecular medicine from the University of Palermo. Then, always at the University of Palermo, she started her PhD in biomedicine, studying cell-to-cell communication in cancer metastasis, mainly focusing on extracellular vesicles. And while doing her PhD, she spent 5 months at the University of Gothenburg. Ornella then moved to Vienna, where she started a postdoc, and she got granted Marie Skłodowska-Curie Actions Postdoctoral Fellowship to pursue a postdoctoral degree. So please help me to welcome Ornella. Ciao Ornella, how are you?
Hello, I'm very, very good, and thank you for this super nice introduction, Mauricio.
Thank you. So let's just start chatting a bit. So first of all, and as I always, probably I'm a bit repetitive with the other interviews, but I think, again, one of the main purposes of this series, personally for me, is to get closer, not only the research that we do, but also the people, so us, that we actually conduct the research. So with this in mind, could you please talk a little bit about yourself? Like, is there anything you want to tell us, to share about your
Yeah,
path,
sure.
about yourself?
I mean, as you said, we are also humans, so we are normal people. So, as other normal people, I have many hobbies. I love cooking, for example, maybe because I'm Italian, I don't know. I love nature in general. I love being surrounded by nature. So, going in a park or especially in the sea. Like, I think my favorite activity is doing snorkeling, maybe because I come from a sea place, so I love everything that is related with sea, the sun, the fishes, and whatever. But to be honest, I think the biggest love of my life is still research. Because to me, doing cancer research is not only a job, it's my passion. So yeah, I would include it among the passions of my life.
That's very nice, yes. I think that also shows exactly the fact that, yeah, we just, at least most of us, or I assume we do this because it's what we like, no? And we chose this kind of topic. And now that just then related to that, is there any like a reason that why you started in the like cancer field, like to do research towards in the cancer field?
Yeah, I must say it's a bit emotional, but I will be happy to share with you and everyone who will see the video. Because when I was a child, I lost my grandmother because of cancer. And when you are a child, I was just 7 years old, you don't understand why a person has a disease and there is no therapy to cure that disease. So when I was little, then I told to my parents, when I will grow up, I will find a cure for cancer. And that was my mission at 7 years old. Of course, now I know that it's impossible to find a cure for cancer. But that sentence that I said when I was very little stayed with me for my entire life. That's why then at the university I decided to study biology, and I was so interested in these DNA mutations, try to understand what is wrong with cancer and how we can find a way to stop it, actually, to find targeted therapies and stuff like that.
That's very nice, and thank you very much for sharing that. Also, because then again, I think it put us in this yeah, human perspective, no? First of all, I think it represents exactly how what we do has an impact in people's life, no? Like you said, like you're working with cancer, but it's also that has impacted also personally, like in my life, like people that I know, no? So it just reflects how important it is and why we do it, no? It's the passion, but also like all the reasons, and at the very end is like for the society, no? So it's very
Exactly.
nice. And thanks for sharing that, because I think it's very important for whoever listens or watches the video. And regarding on the same topic, then, since we now mentioned that you work with cancer, could you please briefly explain like what is your project about, or what, even if it's since the PhD, or what are you working on?
It's different from my PhD topic because before, as you mentioned, I was studying cell-cell communication and in the metastatic process. Well, now here in Vienna, I'm trying to model Ewing sarcoma. So I'm studying the origin of the tumor. That's why also I'm so excited about this project, because I really want to understand what are the very first steps that triggers the tumor. So Ewing sarcoma is a cancer that is very rare. It's more common in children and young adults. That's why I'm working in this Children Cancer Institute or research. And basically, it's a tumor that arises in bone and soft tissues. And till now, it's still a matter of debate, the cell of origin of this tumor. What is known is that it's not characterized by many mutations. It's mostly characterized by a fusion oncogene, which is the result of a chromosomal translocation which fuse together two genes that usually don't have anything in common. But this fusion oncogene basically rewires the transcriptional landscape of the cells and leads to tumor transformation. However, what is interesting is that if you express this fusion oncogene in different cell types, the cell will just die. So it means that to get the tumor, you need the exact specific cellular state or context plus the fusion oncogene. So what I'm doing in the lab is starting with pluripotent stem cells, which you can differentiate in many cell types, and inducing in these cells the fusion oncogenes, which is the known driver of Ewing sarcoma. What will happen is that most of the cells will die because they cannot tolerate the fusion expression, but only the cells with a specific particular cellular context will be able to survive and then being transformed into tumor. And since I'm doing this in a way that is called direct program differentiation, so I upregulate one transcription factor at a time, I will know exactly what were the genes that I upregulate to get the cell state that is able to tolerate the fusion oncogene expression in the end. The final goal is not only find a cell state that can tolerate the fusion, but also push these cells to the full transformation into cancer. And this is what I'm actually doing now in the lab.
Thank you very much. And particular in my case, I think I kind of get it, because I work in something similar, as I told you before. But for the people that is not from the same field or from a similar field, could you now kind of summarize that, and like in words? Because for me also, like, to be fair, those are the words, some of the technical words that I use them every day. So for
Yeah.
me, like, my everyday language. But probably for someone not related to our field, could you just kind of have a quick summary of what you do?
I will try to tell you as I told to my grandma, my other grandma. So basically, let's say there is this tumor that is the Ewing sarcoma, right? Which can arise both in the bones or the soft tissues, like adipocytes or cartilage, this kind of other tissue in our body. The problem of this tumor is that we don't know from where it comes from. Like, if we have lung cancer, we know it comes from the lung cells. If we have colorectal cancer, we know it comes from colon cells. But with this tumor, we don't know. There are some hypotheses, but basically there is still not a good results in terms of that. So to find out from where it comes from, which will help us also to understand better this tumor, I take the pluripotent stem cells, which are like young cells that still don't know what they will do in their life. They can be cells of the skin or cells of the lungs, for example. They can be everything. And I will push them to become everything. And then in these cells, I will add on top something that is specific of Ewing sarcoma to find out in the end which are the cells that can give a rise of this kind of tumor. This is the simplest way I can explain it, probably.
Yes, so correct me if I'm wrong. So basically, what we have is like a stage, no? So the cancer, all types of cancer have an origin, but then sarcoma is already kind of, if not, let's say at the last, but at a late stage. So you take, you try to find what's the origin of it. So because probably here, if you're already too late, it's too late for therapy and for the prognosis of the patients, no? Especially as you said, but they are like normally it affects younger a young population. So by knowing what happens at the beginning, we can try to tackle it from the very start, no? Because as you mentioned,
Exactly.
so these pluripotent cells they have the power to become all, which is that's something I find beautiful about what we do, no? Like
Yeah.
all our cells kind of come from the same origin, and then there are switches that are on and off to become eye or skin or whatever. So you take that cells that are like that can become whatever they or we push them to be, and
Mhm.
then to see where is what's the path that they follow to arrive to the sarcoma, no? And then
Exactly.
so far, do you have like, could you list some impactful things that have come from the project, even like some, I don't know, even if it's a result or like you attended a conference and someone was like, oh, interested in this or to something like that?
So I must say, I'm still in this sensitive stage that I cannot share fully everything I have done in the lab because still I have to do some validation of my results and so on. But like my results so far were very promising because I managed also to reach the part that once I established the model, I injected the cells into immunodeficient mice, I could get tumor. And this tumor also resembled Ewing sarcoma. So this is already amazing. But as you said, like the point is then the therapy, right? Because the strength of the project is that then I will be able to kind of go back and try to rewrite the cancer. Because the moment you have the tumor in the patient, it's already kind of too late to see what happens. Maybe you have additional mutation. It's like so heterogeneous that you cannot really find a target therapy. So I think the biggest impact of this project will be really to say, okay, I did this, and that, and I got the tumor. So now I will be able to go back and like destroy it. As I create it, I can destroy it. That's the goal, basically, let's say. I went to some conferences. Usually people get super excited about the project. I think also the beauty of the project itself is that as well as I'm modeling Ewing sarcoma with this fusion oncogene, you can also think to model other fusion-driven cancers like leukemia, for example, or like other sarcomas as well, which is something I'm also trying in parallel because, I mean, since I have the system, it's always cool to, this scientific curiosity and test different many stuff and so on and so on.
Yes, that's very nice. And there is so exactly to I want you the most difficult part I think is like to find a way, no? And now that you have a way, then you can apply it to different things. So it's very, very nice also, I think, because as you like the way you describe it, no? So to have like now you have a a line that you draw from beginning to the end, and then you can then target even different stages, no? So it's very, very I think even useful and with a lot of potential, no, for like therapeutics, either to prevent or also to then to treat. So it's very, very nice. Also, the way I think what you mentioned about having the mice, because it's like also how normally, at least in our area, works. No, probably you go to you start with cells, but then probably people that are outside the field might think like, oh, this is too far. Even ourselves sometimes, no, like it's too far from finding something. Then you scale it up to like an actual living thing, such as the mouse, which resemble a lot of the metabolic pathways or other things like that, which are very much involved in cancer. So how these things scale up, and every research and every treatment or vaccines that we have started with what we are doing now. So, and with this exciting
Yeah.
ideas, and with excitement that you reflect to us. So,
Yeah,
very
also, I
nice.
mean, I have to mention before jumping into the mouse, we also did some in vitro assays because, it's not that you can go into in vivo experiment without any proof. So there is also this step in between. And like the beauty of working here is that we are physically connected with the children hospital. So eventually we also can get some patient material in the end to compare what I get in the lab with what is actually in the true life in the body. So we always have to remember that we have to stay connected with reality somehow. And we're
Yes.
not just closed in the lab doing our experiments.
Yes, I think it's also very, very, very much important that, and I think it's a great thing that you are that like it's kind of showing also kind of how things should work, no? Like this collaboration with even with the medical doctors, etc. And also the ones that are in close contact with the patients, probably the ones that then people that actually get the samples from them and then that arrives to your hands and all that. So I think it's also like a beautiful way to very clearly present this how science work actually, no?
Yeah.
So it's very, very nice. So then would you say then, because I think you already mentioned it, that like the most innovative or one of the most innovative aspects of your project is this, not to find the origin of this unknown the unknown origin of this cancer. And is there are you already planning like the next step once like if you get this the steps from the original cell to the sarcoma, then what would be the next step, you think?
So yeah, as I say, the beauty would be then to try to go back. So since we know and we can characterize all the steps that will drive to the tumor transformation, find out maybe some like molecular patterns, some molecular mediators, or like something about the chromatin landscape of the cells that we can rewire and drive back, because then the moment we can compare with the tumor, it will be kind of easier, maybe possible to identify some targets for this cancer. Because also the thing is that for this tumor there are not target therapy available, so these children just get this super high chemotherapy and stuff like that, and the side effect can last also for all their life, and it's super sad. That's why it would be good to find good model to try to find new therapies for these children.
Yes. And again, I don't know if I'm being repetitive, but exactly, I think that shows the importance of what the basic research, so that research that is the starting of everything, because so this can the aim of this is to get into a therapy, but for the therapy that probably is what gets closer to like what we get in even in the pharmacies, uh, roughly speaking, started again with all with this with what you are doing. And even though I'm being repetitive again, as I said, I think it's important because it's important for the people that are not working in our field to see that this is how everything starts, no?
Yeah.
So, and it's a lot of work, a lot of passion, but it's very, very nice. So thank you very much also. And I would just want to go back to the very beginning to what you say because I'm curious. So you said that you like cooking. But how easy is it to find like proper products to cook there in Vienna as compare, well, not as compare, because I have
I
lived
mean,
kind of similar situations myself.
I give you an example because I just came back from the Easter holidays and I wanted to do cannoli here. You know, cannoli, it's a sweet from Sicily, like it's this crust part with the ricotta cream and so on. Like here, I would never find a good ricotta. Like it's super hard for me to find like the proper ricotta. But like, except for this, I must say in Vienna, I can find all the ingredients that I usually need. But yeah, there are a few exceptions.
Yes, I can imagine. I have been in something similar, and then sometimes it's like, oh no, you have to adapt. No, but it's very nice.
But this is also part of the game, I think. That's why it's important to live abroad because it really opens your mind. You have to deal also with these small obstacles and find solutions and troubleshooting, which is what we also do in the lab in the end. So
Yes.
it's exciting somehow.
Yes, yes, exactly. That's true. That's also something that it was mentioned last time by Valerio, but it's the same what you just said, no? Like we do kind of research in our everyday life. So it's very, very nice that, and also, yes, I think it's part of, and actually it's the core, no, of the Marie Curie Actions program. So the fact that like this knowledge exchange, so whatever you got from your both previous experiences, both in Palermo and in, it was Germany, no, I think.
Sweden,
In Sweden. Gothenburg
in
was, yes.
Gothenburg.
Yes. And then bring it there back to Vienna to also get the knowledge from them, but also give something from yourself. That's what enriches and that's also something, if not the main beauty of the program, no? So it's
Yeah, yeah,
great.
yeah.
So I also love that. So thank you again. And then just as a final statement, let's say, probably it's too formal saying that, but okay. Could you just please share like a small thought about the importance of research in everyday's life? I know we more or less just talk about it, but I would like just to I like normally to finish with this.
Yeah, we kind of said it, but I think research is the fuel of progress anyways. Like research is super important in everyday life. I think it's because research, it helps to understand how things work. And sometimes things do not work as they should. So if we know how they work, then we can fix them the moment they don't work anymore. And this is what I'm trying to do with my project, right? What is wrong with cancer? What went wrong in these cells? And since I'm trying to model it, I will understand it fully, hopefully, and I will try to fix it. So this is like the beauty of research, in my opinion, to understand and make it better, make our everyday life better.
Yes, I totally agree with you. Thank you for putting it such a beautiful way, and also thank you for being here with us today. So yes,
Thank you. It was a great pleasure, and it was a great pleasure also to meet you.
thank you. Okay, the pleasure is mine. So thank you everyone again, and hopefully you can also have a look if you haven't, if you're curious about the first four videos. Thank you again for joining us, and see you next time to keep digging into what we do as researchers. Thank you. Bye now.